Okay, hello and welcome. It's good to be back. It's Dr. Red Fizzle here, and today I'm going to be talking to you about protein synthesis. So the first step in protein synthesis is a stage known as transcription. So right here, transcription. Right, that's one of the phases. And then the second stage of it is translation. Now, I'm not going to go into too much detail in this particular video about these two processes. If you want to know more about them, then I advise you watch my subsequent videos following this one. And the other thing in relation to like DNA and protein synthesis is the bases. So you've got four bases: your A, T, G, and C. Right. Now these four bases aren't the only bases, but these four bases are the only bases in DNA. DNA stands for deoxyribonucleic acid. I'm not actually going to spell that out for you now because that'll take too long. But anyway, the point is DNA consists of four bases: adenine, thymine, guanine, and cytosine. And there's also RNA molecules. RNA consists of like four bases again, but these are slightly different. So instead of ATGC, it's AUGC. Now the U stands for uracil. It's just one of the RNA bases. And you're probably thinking, why am I telling you the structure of DNA and RNA? What is the relevance of protein synthesis and how proteins are made to DNA and RNA? Well, in order to actually transcribe the proteins, these codes need to be complementary to each other. So what I mean by this is, if I just draw you a DNA molecule here, like, as you can see, these are crossed over, and the reason why this is because of the way they're bonded. There's actually a backbone here. What I mean by backbone is there's lots of different bases like bonded together, and then the actual individual subunits of this, if you like, are called nucleotides, and the bonding down this backbone is known as a phosphodiester bond. Now, a phosphodiester bond sounds rather complicated, but it's not, I can assure you. All that is that's the bond between a pentose sugar and a phosphate group of the next nucleotide. Well, when I'm saying nucleotide, I hope I'm sort of making sense about what one is. I haven't explained it to you yet, so I'm probably not. So you have a phosphate group on the end of one, which is directly bonded to a pentose sugar, which just looks like a pentagon, or is often represented like a pentagon. And then you also have your base in the middle, so draw that as a box. And basically these three subunits make up nucleotides, and this whole chain here are these nucleotides bonded together. Now the phosphodiester bond bonds between this phosphate group and this bit of another one, or the pentose sugar. So I draw you like another pentagon there. I know it's not a brilliant drawing, but the point is that this phosphate group bonds to another one via a phosphodiester bond, as I've just said. So that's basically how these strands are comprised. Now the other thing you need to know is that these strands are anti-parallel. So that means they're just the opposite way up, pretty much. So this one's the 5' end. This end's the 5' end rather of this DNA strand, and the other one has to be the 3' end. If it's not that way up, the bases on here and here won't bond together properly. I haven't drawn this very well because actually these are supposed to be linked in the middle. Like there's a special type of bond that does that, but I'm not going to go into too much detail with the bonding because this video is primarily about how an mRNA strand actually comes in between these two strands of DNA and actually copies the code in order to synthesize proteins. So the next thing I want to talk to you about is, where does this protein synthesis, where does the actual translation phase, or which is the production of the proteins, where does that actually take place in the cell? Well, this takes place in something called a ribosome. Now you should have heard of a ribosome because this video is primarily like aimed at the A-level type level. So you should have heard of ribosomes as a sort of organelle from GCSE. And a ribosome is where the translation phase, which is the second stage of protein synthesis, takes place. So now I'm going to go into a bit more detail with how actually does this mRNA strand slot in between these two. Well, first of all, this needs to be unwound, so it's untwisted by an enzyme, which I'll probably go into detail with at some point, maybe in the next video or something. And once these are untwisted, it's essentially exactly the same, apart from these two strands are straight. And then what you'll notice once it's been untwisted, that these are locking into each other like a zip, these bases. from the individual proteins. Now, what happens after that is something which is quite logical. After this untwists, so it's like rotating and untwisting, it unzips. So these two strands come apart, which creates a gap in the middle. So you end up with something looking a bit like that. And then these sort of bases sticking out here. And then a molecule known as an mRNA can actually squeeze in. Now, there's two strands. There's a coding strand and a template strand. The reason why I'm telling you this is because supposing we're calling this the coding strand, I'll label this C, and this the template strand on the right here, I'll label that T. The reason why I'm telling you that is because the coding strand is complementary to the template strand. It's not identical, it's complementary. Now, I haven't gone into you what are the complementary bases, but a base can only bind to its complementary pair. So you have adenine, thymine, guanine, and cytosine. So if we have a look at these, A, T, G and C. Okay, so A is complementary to T, G is complementary to C. Okay, so you end up with A here, T here, G here, and C here. And what happens is these complementary bases bond together. So this strand, because it's complementary to this, supposing the mRNA strand comes along and collects the correct bases from around the place, and then binds to this 3' end, assuming this is the template strand, it'll also be complementary to the coding strand because the template strand is also complementary to the coding strand. So it's essentially going to be identical to the coding strand because it's complementary to the template strand. Complementary just means that it basically fits into, or the shape is complementary of the particular bases, so they slot into each other nicely. Okay, so that's pretty much the transcription phase done, because what will happen is this mRNA will just go along here, it'll sort of slot its way into here, and it'll find the correct bases that are complementary to the strand, and then it will be transcribed off, which means like taken off, and then it will travel out of the nucleus, which is where this is taking place, via a nuclear pore. Now, pore is just another word given to like a hole, if you like. So P-O-R-E is just a pore, so it's like a nuclear pore, which is just a hole in the nuclear envelope. And we call it an envelope because it's got a double membrane. Okay, so what happens is that this mRNA strand that's complementary to this DNA in the nucleus travels out of the nucleus via the nuclear pore, travels towards a ribosome, which is where the translation phase of protein synthesis takes place. Okay, so in translation, what happens is, once this mRNA strand arrives, it notices that there's a 30S bit and a 50S bit of the ribosome. You don't need to know this in too much detail. I'll label these 50S, 30S. But the main point is this mRNA strand lines up along this 30S strand, and then what happens is it binds to the complementary tRNA molecules that arrive. Because what happens is, around this sort of area in this ribosome, there's these tRNA molecules with a codon and an anticodon. Now the codon is where the actual amino acids bind to. So you've got, I'm not sure, I'll probably turn over the page just to give you a bit of extra space and for me to show you. So you have your tRNA structure here, so it can be something like, they've got a funny sort of structure like this, and then down the bottom here is something called their codon, which is where they have 3 bases which are complementary to one particular amino acid. So we'll just use cysteine for this example, the amino acid cysteine. Okay, and what will happen at this region up here, which is known as the anticodon, is where mRNA strand is. So what will happen is because there'll be like a base here, like at this end, so we'll label this base adenine, for example. What will happen is like a thymine on this mRNA strand, will bind to this adenine at the anticodon, and the reason why it's called a codon is because codon actually suggests that there's 3 3 bases, that make up a codon, because that's what the word codon actually. Well, I don't know what the word codon means, but a codon always represents 3 bases. Okay, so these 3 bases might be adenine, thymine, and guanine. There might be anything basically, but the point is that the 3 bases will be complementary. won't be complementary to the amino acid, but they'll code for a particular amino acid, such as cysteine, for example, I've drawn here. But so once this happens, then what can happen is another tRNA molecule can come around the back, come around here, and could do the same thing, and could be complementary to the mRNA strand here. So it might place another amino acid back here, and this amino acid might be valine, for example. So then what will happen is a bond will form between this valine and cysteine, and I'm not going to go into too much detail with this bond, but it's a peptide bond. You might be able to find a video on that in my biochemistry section if you have a look. A peptide bond will form between the valine and cysteine, and then what will happen is another one will come in, and then it will place yet another one of those, and by this time the first one would have gone, so it'll end up just making a massive chain of these amino acids, which is commonly known as a polypeptide or a protein. Okay, so that's essentially how proteins are formed. You obviously got lots of different types of proteins, such as keratin, which can be found in the skin. Then you have other proteins such as like antibodies, I meant to say, which can be used to fight infection in the immune system. There's lots of different molecules of proteins, and proteins are probably, in my opinion, one of the most important molecules of the body. Okay, so I hope you've enjoyed this video, and that's sort of primarily what transcription is about. If you have any queries about this video or any of the particular details that you think might be wrong, then please sort of let me know. But I hope you've enjoyed this video and hope it's been helpful. So I'll see you in the next video. Thanks. Bye.